Hello, everyone, and welcome to today's APS webinar entitled Neuronal Cell Isolation from Rodent Peripheral and Central Nervous Systems. My name is Jake White, and I will be serving as your moderator today. In today's session, Dr. Rupa Ravishandar and Sam Faraguna will share their research on protocols for isolating and culturing neuronal cells and discuss how various methods support studies of neuronal excitability during nervous system development. Now, before we begin, we have a few housekeeping notes. Out of respect for our speakers, we ask that you refrain from photo or video captures of the presentation in whole or in part. This session will be recorded and available for future viewing through a link on the registration website. Taking photographs of another person's presentation without their explicit permission is prohibited. At this time, all attendees, except for the speakers, are in listen-only mode with their video off. And as moderator, I'll be happy to raise any questions that are entered into the Q&A function located at the bottom of your screen at the end of today's talk. I would also like to thank today's sponsor, Worthington Biochemical, for their support of APS's mission to improve health through discovery. Today's session will end at approximately 12 p.m. Eastern Time. Now, before we begin, we ask that you answer just a couple of questions regarding your experience with cell isolation so we can get a feel for your background. Thank you. Please just take a moment to answer a couple of questions regarding your experience with enzymatic cell isolation and the best way to educate you on these techniques. Thank you very much for taking the poll. It is now my pleasure to introduce our speakers for today. Rupa Ravishandar is a postdoctoral fellow in the Department of Neuroscience at the University of Texas at Austin. Her work examines the interactions between glial cells and neurons during retinal development, using retinal ganglion cells to investigate how glia may influence neuronal excitability through maturation. And Sam Faraguna is the lab manager for the Cantarsi Lab at the University of Texas at Austin. Faraguna's research centers around the peripheral nervous system and the downstream cellular mechanisms of prostaglandin E2. Now, I'd like to invite Sam to join us, and feel free to begin when you're ready, Sam. Thank you.
Thank you, Jacob. Yes, absolutely. Let me get us all squared away and shared and begin. All right, perfect. If y'all can't see this, hopefully someone will tell me. But howdy, y'all. Welcome to the talk. Thank you very much again to Jacob and to APS for hosting this event. We'll be talking today on neuronal cell isolation from rodent peripheral and central nervous system. Yeah, here's a general overall flow of the presentation. I will take you through a brief introduction on neurons, on glial cells at the beginning, and then we'll go into the peripheral nervous system protocol, as well as a little bit of preliminary research that our lab has been doing. After that, Rupa will take it away with the central nervous system. Starting, our nervous system consists of neurons, which are colloquially viewed as the fundamental units that help transmit information. These neurons communicate through electrical impulses by firing what are known as action potentials. Action potentials are an electrical relay of a chemical signal from the sending neuron through neurotransmitters. So as you see in this photo here, that sending neuron is having an electrical impulse go all the way down the axon to the terminal, where a chemical signal, the neurotransmitters, are released into the synaptic cleft, causing that exact same sort of propagation of signal to happen in the receiving neuron. This capacity to fire action potentials is what we refer to as excitability. Importantly, loss of excitability has been linked to disorders including ataxia or ALS. The central nervous system, or CNS as we'll abbreviate it, consists of the brain, the spinal cord, and serves as the main control center for processing and integrating sensory information and directing responses. The peripheral nervous system, PNS, includes all nerves outside of the brain and spinal cord and acts as a communication network, relaying sensory information from the body to the CNS and transmitting those motor commands from the CNS to muscles, to glands, and to sensory organisms. But the mechanisms behind how these neurons gain excitability are still pretty poorly understood. One of the main potential suspects are the glial cells. So glial are these non-neuronal cells that constitute more than half of the cells in the nervous system, and they regulate many important aspects of nervous system development, including myelination and synaptogenesis. So glia originally comes from the Greek word for glue, and they've long been viewed as an unimportant and simply a support cells to neurons. So in the CNS in this photo, you see sort of the microglia, the astrocyte, the oligodendrocyte, and then the precursor for oligodendrocytes, the OPCs. In the PNS, Schwann cells are my main focus. There are a couple other ones that are still somewhat novel, and research is still being done on those. But today we're going to focus a lot more in on the Schwann cells for my part of the presentation. So Schwann cells are the myelinating cells in the peripheral nervous system and allow for the saltatory conduction of action potentials down to neuron. My PI, Dr. Kantarsi, demonstrated in her 2024 paper that Schwann cells were not only sufficient but necessary to induce excitability in developing somatosensory neurons. So how do these Schwann cells influence excitability is sort of our general question in the lab. In her paper, she demonstrated the glial secreted molecule PGE2, prostaglandin E2, is the main driver behind the development of excitability. This molecule is found to increase the total number of voltage-gated sodium channels, which is one of the proxies we use to judge excitability. I'll explain more on that in a second, but that's sort of one of the ways we're able to research, or one of the ways we're able to understand PGE2's effect and glial's effect on excitability. So you can see from this schematic here, you have Schwann cells, which are individually wrapping segments of the axon and myelinating segments of the axon and secreting this factor PGE2 that has this direct influence on the excitability of this neuron. So let's look at something that has been torturing me for a while, which is sort of the role of PGE2 in glial-induced excitability, and more so sort of the downstream mechanisms of PGE2. A lot of current research is still being done on the downstream mechanisms, and this is really just one theory, and a very simplified theory, onto the intercellular processes that influence or that are cascaded from PGE2. So EP2 and EP4, PGE2, this molecule binds to EP2, EP4, which are G-coupled protein receptors, causing that very classic intercellular cascade, leading to an increase in cyclic AMP. An increase in cyclic AMP drives sort of PKA's phosphorylation of downstream targets, such as CREB. CREB is a transcription factor that binds to DNA, causing the transcription of genes such as the voltage-gated sodium channels that I had mentioned before. So how are voltage-gated sodium channels and excitability related? So if you take a look at this this little chart here, which is an action potential pretty much, and labeled are different points in the action potential. So looking at that very initial depolarization, that is governed by the opening of these voltage-gated sodium channels. So voltage-gated sodium channels endow these neurons with action potential firing abilities. They initiate this depolarization of the neuronal membrane by opening up, and an increase in the number of these channels would lead to a more excitable neuron, one that can more readily fire these action potentials. So from what we've briefly covered with PG2 and glial interaction, this leads into our protocol, which allows us to properly answer the above question of do glia initiate DRG neuron excitability. So this is just a brief schematic of what we do, and I'll go into this obviously in a lot more detail. Something that I do want you to note is we're isolating these neurons from embryonic day 15, so E15 rat pups, or rat embryos, I should say. At this point in time, DRGs are post-mitotic, but they have no spontaneous activity. This is also before the maturation of Schwann cells. So before non-myelinating and myelinating Schwann cells can have that sort of influence. So we're isolating these cells before they are excitable. So that's just an important thing to hammer home. So yes, the DRG isolation protocol here is, again, another one of those brief, maybe a little more in detail schematics. This is coming from a paper that we just published, myself and one of the amazing research assistants in our lab, Karina Pena. So let's get into it, starting with the dissection. So here is a simple, all right, so what we're doing here is, again, taking these E15 rat embryos, and what we're trying to do is we're trying to remove the spinal cord and then dissect away these DRGs. So these next few photos are going to be what we see under the microscope as we're doing this protocol. And this first one here is a rat embryo after it had been removed from an embryonic sac. As you can see, looks very much so like a small rodent would. And what we're wanting to do is we're wanting to trim away a lot of this excess tissue and expose the spinal cord. A huge part, and something that I'll stress time and time again, is that what we want to do in this protocol is trying to create as pure of a culture as possible without damaging the neurons. So we're wanting to trim away until we get to what you can see here on the side are these DRGs, these little oval shaped things. These are the DRGs that we're wanting to isolate. So they're still attached to the spinal column, but what we're going to do is we're going to pluck those off pretty much and create, again, trying to minimize as much contamination as possible through these surgeries. After this point, after we finish all the surgeries on all the pups, what we start is the enzymatic and mechanical digestion. So again, we're trying to create a single cell suspension without damaging the neurons. What we do is we use papain to break apart the extracellular linkages between cells, and we use DNase, strangely enough, to break apart any DNA escaped from dead cells. So you're bound to have some dead cells, you're bound to have some sort of DNA leakage, and DNA itself is very sticky, strangely enough, and will cause cells to clump together. So we're using DNase not to alter the, or not to influence any of the DNA inside living cells. This is for dead cells, which causes this clumping together. And the low ovo suspension here neutralizes the papain and DNase so as not to over digest the cells. Again, trying to keep these cells as healthy as possible while creating a single cell suspension. Right. So this is just another schematic of papain itself. This is a simplified version of its process of these polypeptide fragments and the creation of these polypeptide fragments. Now we're going into the more novel section of this protocol, which is the immunopanning plates. Once we have our single cell suspension, it's time for the immunopanning, right? And immunopanning uses primary and secondary antibodies to isolate different cell types. So for my prep, I use two negative panning plates to remove, the first one to remove blood and endothelial cells through BSL1. And the second plate is to remove contaminating glia through the CD9 plate. BSL, again, is going to be this primary antibody here, or in this diagram, this schematic, that magenta color. And what it's doing is you're able to create these plates that can selectively take a cell type of your choice and secure it on as you wash off the other cell types, or in our case, take the suspension that is left behind and use that in further isolating plates to make sure that you have as pure of a culture as possible. So later on, Rupa will show an example of like a positive planning plate. So you can either discard the solution or keep the solution that is left behind. So here's just a simple schematic of our BSL and CD9 plate. Again, so the BSL taking these blood cells and the CD9 taking these contaminating glia, hopefully leaving behind this perfect DRG culture. So here is a couple photos of our DRGs after the first day and after day 6. You might see a little bit of cellular debris, which is pretty unavoidable. I mean, you're doing a pretty aggressive treatment. But by and large, these cultures are virtually uncontaminated and are up to 95%, 97% pure. Sometimes we also do FUDR treatments. FUDR kills off any cells that are or arrest any cells that are dividing cells. So stopping the synthesis or the synthesis of new DNA. But again, these are some of our cultures that we start using. But now the question sort of turns to, once we have these pure cultures, what can we do with them? So the 3 most common methodologies we use in this lab are going to be RNA-scope, viral infection studies, and then also co-culture experiments. Starting with RNA-scope, this procedure fluorescently labels mRNA molecules. What we are interested in is the rate of transcription of 3 sodium channels in DRG neurons, the N of a 1.6, 7, and 8. And again, as I've mentioned before, we're looking at sort of the changes in these sodium channel expressions as a proxy for excitability a lot of times. As you can tell from these figures here, there's a significant increase in the number of mRNA transcripts for DMPGE2 conditions compared to control and compared to CREB inhibitor. We use the CREB inhibitor to demonstrate the transcription of these genes does in fact fall under this transcription factors domain, an area of influence. DMPGE2, I should mention, is a dimethyl version of PGE2, so it's a more stabilized version of PGE2 and will allow for a more consistent chronic treatment, as you would see in actuality in real life. As a Schwann cell wraps around it, you would have that sort of continuous treatment of PGE2. So that's what DMPGE2 tries to mimic, while as PGE2 usually breaks down a little too quickly. Moving on to our viral infection studies. This next technique is for looking at calcium, and we're using a genetically encoded calcium indicator, this little Wi-Fi password here, JGCamp8F. Calcium infection allows us to capture action potential firing in real time. The genetically encoded calcium indicator fluoresces when calcium binds to it. This indicator has very, very fast dynamics that allows us to detect multiple action potentials occurring in the cell. And if you look at these graphs that we've created here, the first one is just showing from the camera that we're using, the capturing of these action potentials of this fluorescence that we see. Control and DF is change in fluorescence. So we're looking at fluorescence over time here, and you're seeing the ability to fire these action potentials in DMPGE2 and in the cyclic AMP condition. And if you cast your mind back again to that sort of downstream mechanism graph for DMPGE2, cyclic AMP is part of that pathway. So again, we're just trying to show that, trying to piece together this pathway as a whole. And again, these cells are actually are stimulated, so this is not spontaneous activity. This is activity once stimulated. The next one we use is a voltage, a genetically encoded voltage indicator. So voltage is for sodium channels, and this sodium channel GTP actually fluoresces when the membrane depolarizes. So it's the opposite of the calcium. So it'll get dimmer as the action potential happens. But either way, you see a very similar sort of graph here from our data where DMPG2 causes such a significant increase in spikes compared to the controls. And again, this is very preliminary data for us, but yes. Lastly, for the viral infection studies, we've started using this technique called Brainbow. So Brainbow, um, this is a fun one. It's essentially you're fluorescing individual neurons and axons in different colors, which allows us to visualize again these separate neurons in culture. As you can tell, we still haven't fully perfected the technique, but it's certainly getting better and better. This is used primarily in a lot of our regeneration work that we've started doing. So what we'll do is we'll culture these neurons, sever axons, and then treat with different conditions like DMPG2 and visualize sort of this regrowth. There's a lot of work that we're starting to do with DMPG2's role in regeneration. So as you can tell, just as a baseline, there is a lot more growth for the DMPG2 condition than otherwise. Finally, looking at our coal culture experiments. So our coal culture experiments are also pretty new to this lab. What we're doing is we're taking these peer cultures of DRGs and we're plating them with Schwann cells isolated in a different prep. And the overall goal of this is to see if Schwann cells and myelinating Schwann cells are able to influence the development of subtypes of neurons. So when we isolate E15 DRG neurons, they haven't yet separated into subtypes. So you can imagine subtypes as 4 sensory neurons. There's neurons that will sense pain, hot, cold. You'll have mechanosensory neurons. You'll have proprioceptive neurons. So there are, while these DRGs are certainly committed to the neuronal fate, they are not yet at their end destination. And we're trying to figure out if direct contact with myelinating Schwann cells, with unmyelinating Schwann cells, seeing at what point and under what conditions did these subtypes start to develop. In fact, like if you keep these pure cultured E15 DRG neurons for as long as you want, or as long as we've left them in culture for close to 30 days, they do not develop any sort of subtype. So we're trying to see what factors influence that final stage of differentiation. And here's again some brief research that we've done into promoting the neural subtype development. Paraalbumin is a marker for proprioceptive neurons, which proprioception is sort of the our perception of our body's time and place in space, I guess. And it's associated a lot with balance and movement issues and disorders. So what we can see from here is there is a not significant, but there is a trend towards Schwann cells being, or Schwann cells influencing the subtype development and subtype specificity. Oh well, essentially allowing for Schwann cells to or Schwann cell culturing helps along that pathway of going towards proprioceptive neurons. So as a whole, what I've hoped to show today is that sort of negative panning allows for pure neuronal cultures for DRGs. Pure cultures, once we have these, allow for isolated studying of the development of excitability neurons without glial interactions. And PG2 and Schwann cells promote excitability and regrowth in DRGs with the potential to fuel subtype development. And at this point, I would love to hand it off to Rupa. So I'm going to stop sharing and stop talking.
All right. Hi, everybody. My name is Rupa Ravishandar. I hope everyone can see my slides all right. My name is Rupa Ravishandar, and I'm a postdoc fellow at University of Texas at Austin, also in Dr. Husni Kantachi's lab. And today I'm going to take you all through the second half of the webinar about how we can study neuronal excitability in the central nervous system. So in this talk, I will be describing to you the protocol for isolating retinal ganglion cells. I'll also introduce to you some background on why neuronal excitability is important in the central nervous system, and then some preliminary data following that. So retinal ganglion cells are the neuronal cells in the retina that are present in the ganglionic cell layer. The retina itself is a multi-layered tissue with photoreceptors, bipolar cells, as well as the retinal ganglion cells. The photoreceptor cells actually detect the light. They transmit the synapse onto the bipolar cells. This information is then transmitted into the retinal ganglion cells. And these axons then project through the optic nerve into this lateral geniculate nucleus and then into the visual cortex, thus enabling us to process visual information. One of the advantages of retinal ganglion cells is that it's easily accessible. In fact, the retina is actually an extension of the brain, and embryonically they originate from diencephalin, which is part of the forebrain, and the retina itself has a lot of similar features, such as glial populations that are surrounding and that are supporting these retinal ganglion cells, thus making this as a very unique model to study CNS neuron function. To introduce to you why this neuronal excitability is important, I would like to highlight one of the most influential and pioneering work by Dr. Ben Barres and colleagues, wherein very initial studies, which was back in 2002, showed using these retinal ganglion cells that by these RGCs by themselves actually do not extend axons. However, with these with addition of electrical activity along with neurotrophic factors, these retinal ganglion cells were able to extend or grow axons and also aid in RGC neuron survival a little better. And supporting this, there's this panel on the right here showing how they conducted this using multi-electrode array systems, where the RGCs or retinal ganglion cells were cultured, and then following stimulation along with these neurotrophic factors, you see this increase in axon growth, suggesting that this was very critical, or physiological levels of electrical activity is actually necessary for axon growth and survival. We do see in a number of different diseases, as my colleague Sam had described, such as ALS and ataxia, where this imbalance in excitability can lead to these neurodegenerative diseases. And indeed, in one of the retinal degenerative diseases called glaucoma, you do observe differences in excitability in earlier stages. The panel in the bottom here shows a glaucoma model called the microbead occlusion model, wherein it increases the intraocular pressure, and you do see 2 weeks following this injury, you do see this increased levels of excitability, and then this reduction of this excitability. So typically in some of these retinal degenerative diseases also, these are characterized by hyper and hypoexcitability phenotypes. When do these neurons actually become excitable? Neurons actually become excitable during the course of development, and for this excitability to occur, there needs to be expression of voltage-gated sodium channels. These are fundamental to firing action potentials. Since we will be looking more to the retinal ganglion cells in this talk, I would like to talk a little bit about the emergence of excitability in retinal ganglion cells. In the panel on the right, what you see is that these retinal ganglion cells attain full mature firing patterns by postnatal day 25. They actually start off with only a single action potential at P4. To look at it a little more in detail, you see that in this timeline, you observe that the retinal ganglion cells actually are generated as early as embryonic day 11, and they keep generating until birth. This is followed by a lot of different time points where the glial cells are arriving. And as I described before, at postnatal day 4, the RGCs are only firing a single action potential. And over the course of this development towards P25, they achieve this mature or 100% firing patterns by P25. Intriguingly, this period of this emergence of excitability is also coincidental with the arrival and maturation of many of these glial cells, particularly myelination of RGC axons by oligodendrocytes that happen in between this period where this excitability emerges. And so this led us to ask what actually promotes neuronal excitability during development. And one of the questions that was intriguing to me based on previous work by Dr. Kantarci, where Schwann cells in the periphery were critical for inducing somatosensory excitability, this led me to question if glia could induce neuronal excitability in the central nervous system as well. And as described by Sam earlier in the central nervous system, these are surrounded by a lot of by glial subtypes, oligodendroglia, astrocytes, microglia. The oligodendrocytes help in ensheathing these axons to enable faster signal transmission. So one of the earlier work, again by Frieger and Ben in Dr. Paris, showed that co-culturing glia with these retinal ganglion cells, in fact, increased the frequency and amplitude of excitatory postsynaptic currents in retinal ganglion cells, as shown over here. And so this led me to question what these factors actually were. And this was something that wasn't, that is still a little unclear. And so one of the ways that we are trying to answer this question is by isolating RGCs and away from the glia by this protocol, by this immunopanning procedure, which is very similar to what Sam described earlier. To do this, we isolate the retinas from P1 to 3 rat pups. Typically in a litter, we get about 10 to 12 pups, and then we extract about 20 retinas. And we remove any vasculature. And following this, we treat the tissue with papain for digesting the tissue. And this tissue is then, it's important while digesting, these should be treated with 95% oxygen and 5% CO2. Giving this bubbling enables better viability as well as stabilizing the pH. It's important to optimize this level or the concentration of papain that we use such that you don't overdigest or underdigest the tissue. Following this, to stop the digestion process, we use low OVO as it was described earlier. And this brings the trituration process itself enables the single cell suspension. And then following this, we perform immunopanning, which is the first part is the negative selection using BSL, which is coated on these immunopanning plates. We throw this heterogeneous population of cells onto this BSL plate. BSL particularly captures microglia, macrophages, or endothelial cells, which we don't care about. Whatever remains from this cell population, we move on to the positive selection, which is immunopanning plates, which are coated with Thi 1.1 antibody. And what this does is it captures the retinal ganglion cells that we care about, and we culture these retinal ganglion cells in growth factors such as CNTF, BDNF, and forskolin. So some of the most critical steps for a good RGC yield are, as I mentioned before, papain is the digestion step, which is very critical. You don't want to over-digest or under-digest the tissue. So typically for about 20 retinas, we use 50 units of papain, which is pretty sufficient since the tissue is pretty small. And then it's also important that you give this, during digestion, this 95% oxygen and 5% CO2 helps with better viability. The trituration process itself needs to be done very slowly, such that you're not completely dissociating in one go. It needs to be very slow for better cell viability. And the third part is the antibody that is being coated. You want to make sure this antibody generated from this hybridoma is concentrated. We typically use a 50 kilodalton filter to concentrate this antibody from this hybridoma and then use them for panning or for selecting for RGCs of higher purity. So this is how the RGCs or retinal ganglion cells look one day post-seed. You see them in this bright field image where they are projecting axons, and this was also post-fix. They were stained with this neuronal marker NUN, and thus showing highly pure cultures. One of the things that, as I mentioned before, one of the most important question that we are interested in is to know whether glial cells induce or influence neuronal excitability. And so to do this, we also have optimized an immunopanning protocol to isolate glial subtypes such as astrocytes and oligodendroglia. And we do this by using positive selection by this marker called as ITGB5 for astrocytes and O4 for oligodendroglial lineage cells. And post panning and culturing, for validation, we use OLIG2 as a marker for oligodendroglia, and GFAP, a marker for astrocytes, here in red, suggesting that these are highly pure glial subtypes following immunopanning. So here's some preliminary data wherein these retinal ganglion cells were treated with oligodendrocyte-derived conditioned media. And what we do observe, as Sam mentioned earlier, that one of the metrics that we use to measure excitability is this expression of voltage-gated sodium channels. And we observe this following treatment with oligodendrocyte-derived conditioned media, you see an increase in the expression of NEV 1.2. which shows that there's something that's secreted from this conditioned media that enhances the expression of voltage-gated sodium channels. We were then curious to know whether this could be, as seen previously in the peripheral nervous system, could PGE2 be that factor that promotes RGC excitability. And so to test this, we treated these immunopanned retinal ganglion cells with the stable form of PGE2, DMPG2, and we observed the voltage-gated sodium channel expression, and observed that following treatment, we do see an increase in NAV 1.2 and NAV 1.6 expression. These were conducted by RNA scope studies. And we also see an increase in NUN, suggesting that these RGCs are becoming more mature following DMPG2 treatment. Another way we measure excitability is the use of an ASAP5 virus, which is an optical reporter for membrane voltage changes. This was developed by Dr. Michael Lin's lab in Stanford University. And essentially, after infecting these cells with this ASAP5 virus, they develop this kind of GFP expression, as you can see in this image over here. And what we did observe is that following DMPG2 treatment, we observed this increase in the number of firing action potentials per neuron, indicating that PG2 treatment does increase the excitability in terms of in live cells, in terms of firing action potentials or firing patterns. Another thing that we were interested in was to look at whether PGE2 treatment actually increases axonal growth or survival of RGCs. And to do this, we utilized this live death staining using calcine AM and Bobo-3 iodide. And following DMPG2 treatment, we observed this increase in the percentage of live cells and also an increase in the neurite length at a 100 nanomolar dosage. And this indicates that potentially DMPG2 has abilities to probably regenerate axons and may have some kind of therapeutic potential in some retinal degenerative diseases, which I'm planning to test in the future. So in summary, I hope today I've been able to convince you guys that retinal ganglion cells are an excellent CNS model to evaluate various aspects of neuronal excitability, firing patterns, and axon growth. I also showed you an immunopanning protocol to isolate our retinal ganglion cells away from glia. We also looked at how the molecule PG2, previously shown to induce sensory neuron excitability, can also induce excitability in the central nervous system, such as retinal ganglion cells. And PG2 also enhances action potential firing in RGCs and promotes RGC axon growth. So with that, I would like to thank all the members of the Kantarchi lab, in particular our mentor, Dr. Husni Kantarchi, has helped us support and develop this project and guide us through this through these months so far. And thank you all. At this point, I think we can take any questions.
Thank you so much, Rupa and Sam. Feel free to join us. We have quite a few questions and more questions coming in for you. So we'll take as many as we can. This is for either of you. The question is, I have a quick question about hyperpolarization. What does it mean when you have steep hyperpolarization curve? What influence does it have on K efflux?
Yeah, I can take this, Rupa, unless you want to take it. But so for those who may not know, hyperpolarization, when you look back at that sort of action potential curve that we see, so sodium channels open, peaks, and then comes back down. That hyperpolarization is what happens when it actually the membrane potential dips down even further than where it started at. This is called the refractionary period. Hyperpolarization, so that's usually a sign of an increase in potassium efflux. If the question is, what does it mean? Like that is actually, it's a beneficial thing to have, that refractionary period. So during that period, it's a lot harder for neurons to fire. So you want that, so there's not like a tonic that happens where you're just like continuously firing a neuron. So that potassium efflux is good and normal. When it comes to like even more hyperpolarization, I can't speak to that too much, but I'm sure that's a sort of an issue with some sort of secretion of these ions. Like that balance is very, very key to have. I don't know if you want to ask or put more onto that, Rupa, but
yeah, I think I agree with you. Um, uh, that refractory period or the extension, I mean, the hyperpolarization is typically a membrane potential is for RGCs is like the resting membrane potential is -70 and then goes all the way to +50 millivolts. And then that hyperpolarization is that characterized by that point where it goes even below than -70. And yeah.
yeah, and again, that's a common thing to see, and in a lot of cases, a beneficial sort of break for these neurons to reset.
Thank you
But
for
if it's
clarifying.
yeah.
The next question, and I believe this is for Sam as it is protocol based, but Rupa, please also, feel free to answer is, will this protocol work for adult DRG isolation? If not, do you have any recommended protocols for adult DRG isolation?
Yeah, so our protocol in particular will not work for adult DRG neurons for a few reasons. First, our surgical technique, you'll have to change the dissection. Um, what happens when you start doing DRG isolations on adult mice or adult rats, that the I mean, like when you're cutting an embryo, it's a lot easier, it's a lot softer of a tissue. So you're gonna have to deal with bones, deal with a lot more connective tissue in the past. So you'll have to change the surgical technique. The second thing is our papain is not going to be strong enough to you'll have to change how you disassociate tissues from each other. Papain is not going to work for that. There are some protocols, and I don't know them on the top of my head, but I know in our paper that we published, we do mention some of these adult DRG isolation protocols. I'm sure I can send that over to y'all at some point if we get connected. Um, but they use I can't remember the exact chemical. I think they use some sort of trypsination to help with the to help do exactly what Pape does for us for the disassociation. And also, a lot of times adult DRGs don't last as well and don't live as well in culture. But that is by no means something to be a deterrent. That just means that like whatever concentrations we use for growth factors just might have to be tweaked a little bit. So there's room for tweaking.
And the next question says to Sam, thank you for your lecture. Have you ever used adult rodents for neuron/glial cell culture? If so, is the procedure the same? So it's similar
Yeah,
question.
yeah, same idea. For glia, we do glia isolation on pups, so that's a lot easier of a protocol when it comes to adults. But again, same point for DRG isolation as before. You have to tweak it. You'd have to tweak it a bit, but there are some protocols. Again, I just can't remember them off the top of my head, but there are some published protocols out there for adult DRGs.
Uh, are there any detailed isolation protocols with positive and negative selections that you may share?
Uh, I mean, Rupa, you just gotta Rupa can publish, yeah, yeah, then maybe, but I don't know. Rupa, do you know of any? Uh,
I mean the one that the one that I have experience with is the RGCs where you use the negative panning plate, which is BSL, and then you use the positive plate of Thai 1.1 for selecting RGCs. Um, uh, was there anything else that the question specifically asked?
No, no, just a request to share any more detailed isolation protocols that had both positive and negative selection steps.
yeah,
Um,
not many that I know of.
when on that same topic, we have another question of what dilution was used for CD9 immunopanning?
The dilution for CD9, you're 90 microliters into about 10 milliliters, whatever that dilution is off the top of your head, if you can do that. Um, so that also changes a lot, I will say. So um, that is one of the few things that you just have to sort of tweak as you go along. As we mentioned with papain as well, there's some moving parts to this protocol and to a lot of what we do. So there'll be. I remember when I first started doing this, we were doing two CD9 plates as well, just to try like looking at your cultures, you can sort of tweak these techniques. Um, it the the one thing you have to remember is you are kind of restricted by your plates as well. Like you can, if you don't have enough secondary antibodies, if you add a lot of the primary antibodies, it's not going to do much. It's going to exhaust the secondary antibodies that are already there. So you're sort of bound between these two conditions and also the size of your plate. Um, so yeah. Sorry about that. But all of that is certainly in our paper, in our protocol.
Been able to do the next question, which is, have you been able to do neuronal scRNA sequencing with glial interaction versus without it?
Rupa, do you want to speak to this?
Yeah, I think that's a great question. I mean, it's something that we are really very much interested. One of the things that we are trying to do is collect this glial condition media, and then further look at factors specifically, and also after treating these retinal ganglion cells with the conditioned media, what actually happens to these RGCs in terms of expression. So that's something in the docket, but not as of now, but definitely in the future.
Yeah, one of our like prefrontal cortex camps. So we have 3 camps in this lab: the peripheral nervous system, RGCs with Rupa, and then we have another section which is the central nervous system, prefrontal cortex, and they've just done single cell RNA-seq on this exact topic. So treatment, they look more for the treatment of astrocyte conditioned media to these neurons versus without. So that is certainly something that we've been looking at.
Thank you. There's actually 2 questions regarding, the material that coats the plates for
Mm.
two different steps of your methodology. So first question is, do you use specific material coated culture plates for negative or positive immunopanning? And then also, what about for culturing the cells?
Yeah, I can speak to that again. So for the immunopanning, there's not any sort of specific coding outside of the secondary and then primary antibodies that we're using. Rupa, unless you would push back on that, I think we're just using regular petri dishes.
Yeah.
And
Yeah, and then
yeah,
it's important that you use suspension dishes, not the tissue treated dishes.
yeah,
And typically for panning, it's just the secondary followed by the primary, and like for BSL, I guess it's just BSL that goes on.
right. But for the cover slips that we use for culturing cells, so we culture on these. I mean, we've different size well plates, but a lot of times 24 well plates is kind of what we use. So we have these glass cover slips that are coated with PDL lysine, and then we also use laminin on top of that as well. So those are coated. Those are all in-house. We wash these glass cover slips for a few months beforehand just to make sure that there's no contamination. And then the day before we do that coating step. So yes, for culturing DRGs, for culturing RGCs, for culturing your prefrontal cortex neurons, yes, yeah, that's important.
And this is actually a question. Thank you for that. This is actually a question that kind of goes back to immunopanning and potential other completely different techniques. And the question is essentially, would flow cytometry actually be a better alternative?
Ruba, do you want to take that one?
So I mean, we haven't tried flow cytometry for the retinal ganglion cell isolations in particular, but I mean, flow cytometry could be a bit harsh on the neurons. I mean, you don't, since it's going through, I mean, through the sort machine, it might probably um, maybe it is a bit harsh in terms of the processing, whereas the plates are more like you're just transferring rather than it going through a very small nozzle. So I might expect maybe the immunopanning protocol is a little less harsh in that sense. But again, like I've not tried a flow cytometry or a flow sort protocol, so yeah.
Appreciate that analysis. And this is a fairly simple question. What kind of slides do you use? And I believe they're speaking to the brand when when you're cold culturing DRG neurons.
So the slides we use, I guess that it means the cover slips we use. So what we're plating these on.
Yeah.
Those are on the Carolina Bio, I believe, is the cover slips we use for this. Again, that's in our protocol. I'm pretty sure that's right. That's the manufacturer that at least comes to mind initially when we order 1 million of these things, so it's always somewhat embedded in my mind during all of our ordering. But yeah, I think Carolina Bio, the I don't think the thickness matters at all, but that matters more for what kind of microscope you're using and what kind of imaging you'll be doing. But yeah, that those are important, and again, very important to wash these with ethanol for a long time, and also very important to pre-coat with your PDL and your laminin.
Thank you. And there's a couple of more broader questions, since we do have the time.
Yeah,
What are the possible therapeutic applications for PG2?
yeah, that's a very good question. So I can give a little peek behind the curtain of what we're doing. So we've just started a sciatic nerve regeneration protocol. So looking at, can PG2 treatment with PG2 induce sort of this regrowth and for severed and crushed axons. So I think that's, again, we're early stages in all of this, but looking at PG2 as a cofactor for inducing regeneration is going to be a big part of what we do moving forward. And then for Rupa,
Yeah, I think as Sam said, a similar kind of a model that we would like to test it on is like an optic nerve crush assay, which mimics, I mean, it's an injury model for glaucoma or any kind of RGC degeneration, and this enables us to, we are planning to do like intravitreal injection of PG2 and see if this can enable axon regeneration. We have some good preliminary data from just an in vitro process of treating these RGCs with PG2, that we were able to see axon growth and neurite length increase following this treatment. And so we are hoping that maybe PG2 could have some therapeutic value in terms of retinal degenerative diseases like glaucoma.
I appreciate that. And actually, we have a follow-up question that was just submitted related to this. Do you only use a crush model for sciatic nerve regeneration studies?
as of right now, that's what we're going with, but we'll branch out depending on the results of it all. Um, so yeah.
And then one last question, actually, apologies, going back to the methodology. How long do you let the brain lysate sit on the immunopanning dish during cell selection? And is this at room temperature, or is this, in a 37°C incubator?
Yeah, for retinal ganglion cells, we typically keep the cell suspension in these panning plates. For the BSL plates, it's about 30 minutes-ish with a little bit of shaking in between 10 minutes. And this is done at room temperature. For the positive selection also, we do around like 20 minutes shake and then another 20 minutes. This is also done in room temperature. So we have, for the coating part the day before, when we do the secondary and the primary, the secondary is kept in once we coat these panning plates with the secondary antibody, that goes into the 4° overnight. And then the next day we do a couple of washes and then throw the primary antibody on. That primary antibody panning part happens in room temperature for like at least 3 to 4 hours. So that's kind of the protocol that we follow.
All right. We have one more question. And what is the role of knockout mice in this experimental paradigm?
Yeah, so we've developed a few different strains of either knocking out PG2 in, I guess, a whole bunch of different cell types. But yeah, we do not only cell culture work, but we do a lot of animal models as well, just trying to branch out. And confirm what we're seeing in culture, because we know in cultures is rarely or is not always applicable to exactly what's going on inside of an animal and inside of a human.
Yeah, so we do have like the enzyme that helps with the synthesis of PG2 is the PTGES3. So one of the things that we are looking at is conditionally knocking it out in specific cell types. Since we suspect glia are the primary producers of PG2, we are looking into like oligodendrocytic knockout of PTGS3, and then assessing how development of RGCs are affected by it, and same with an ALDH knockout mice to in ALDH PTGS3 being knocked out in ALDH Cree for astrocytic knockout to look at what happens if you ablate specific glial subtype derived PG2, and then looking at various aspects of development or excitability of these neurons. So that's something that we are very much interested in, are pursuing a lot of research on that aspect.
Exciting extension of your work. We're actually about time now. So I just wanted to thank you both, Rupa and Sam, for taking the time to present your great work. Want to thank again Worthington Biochemical for their support of this program and APS in general, and everyone else, all the attendees today for joining us. There is a brief survey we'd love for you to complete about today's session, and also about future topics in cell physiology that you're interested in seeing us present. Again, thank you all for joining us, and have a great day. Take care.